Carbohydrates, fats, and proteins can all be degraded to release energy, but carbohydrates are the most readily used energy source. When we eat a meal, we are eating quantities of these nutrients that will provide the energy required for life processes. We can organize catabolic processes into 3 stages, which are summarized in the figure on the right. The purpose of the first stage of catabolism is to degrade large food molecules into their component subunits. These subunits, simple sugars, amino acids, fatty acids, and glycerol, are then taken into the cells of the body for use as an energy source. Polysaccharides are hydrolyzed to monosaccharides. Salivary amylase begins the hydrolysis of starch in the mouth. In the small intestine, pancreatic amylase further hydrolyzes the starch into maltose. Maltase catalyzes the hydrolysis of maltose, producing 2 glucose molecules. Similarly, sucrose is hydrolyzed to glucose and fructose by the enzyme sucrase, and lactose, or milk sugar, is degraded into glucose and galactose by the enzyme lactase in the small intestine. The monosaccharides are taken up by epithelial cells of the intestine in an energy-requiring process called active transport. The digestion of proteins begins in the stomach, where the low pH denatures the proteins so that they are more easily hydrolyzed by the enzyme pepsin. They are further degraded in the small intestine by trypsin, chymotrypsin, and elastase, and other proteases. The products of protein digestion, amino acids, and short oligopeptides, are taken up by the cells lining the intestine. This uptake also involves an active transport mechanism. Digestion of fats does not begin until the food reaches the small intestine. Fats arrive in the duodenum, the first portion of the small intestine, in the form of large fat globules. Bile salts produced by the liver break these up into an emulsion of tiny fat droplets. Because the small droplets have a greater surface area, the lipids are now more accessible to the action of pancreatic lipase. This enzyme hydrolyzes the fats into fatty acids and glycerol, which are taken up by the intestinal cells by a transport process that does not require energy. This process is called passive transport. A disaccharide can be hydrolyzed by a glycosidase, which breaks a glycosidic bond to produce 2 monosaccharides. A peptide is hydrolyzed by a peptidase, which produces 2 amino acids, and a lipase hydrolyzes the ester bonds in a triglyceride, yielding fatty acids and glycerol. The monosaccharides, amino acids, fatty acids, and glycerol must now be assimilated into the pathways of energy metabolism. The 2 major pathways are glycolysis and the citric acid cycle. Sugars usually enter the glycolysis pathway in the form of glucose or fructose. They are eventually converted into acetyl coenzyme A, which is a form that can be completely oxidized in the citric acid cycle. Amino groups are removed from amino acids, and the remaining carbon skeletons enter the catabolic processes at many steps. Fatty acids are converted to acetyl coenzyme A and enter the citric acid cycle in that form. Glycerol, produced by the hydrolysis of fats, enters energy metabolism via glycolysis. Acetyl coenzyme A carries acetyl groups, 2 carbon remnants of the nutrients, to the citric acid cycle. Acetyl coenzyme A enters the cycle, and electrons and hydrogen atoms are harvested during the complete oxidation of the acetyl group to carbon dioxide. Coenzyme A is released or recycled to carry additional acetyl groups to the pathway. The electrons and hydrogen atoms that are harvested are used in the process of oxidative phosphorylation to produce ATP.